Hello. Today we're going to be talking about yet another important metabolic pathway, the citric acid cycle. The citric acid cycle is a series of 8 reactions, most of which are amphibolic. It is also known as the Krebs cycle or the tricarboxylic acid cycle, which is shortened to TCA. Many of its intermediates are used in different anabolic pathways as building blocks. Also, many different catabolic pathways will feed into the citric acid cycle at different locations. The goal of the cycle is to both oxidize acetyl-CoA to generate reduced electron carriers, and also to regenerate oxaloacetate so the cycle may continue. This cycle also prevents the creation of methane while oxidizing acetyl-CoA. 2 carbons enter as an acetate molecule, and 2 carbons leave throughout the cycle as carbon dioxide. Acetyl-CoA is an activated acetate molecule. Seen here, the molecule on the bottom, the acetate, is in blue, which is generated from previous metabolic activity. Coenzyme A is in black. Coenzyme A is an excellent leaving group and is a modified ADP molecule, which is additionally phosphorylated at the 3' hydroxyl group, dedicating the nucleotide for usage as coenzyme A. Pantothenic acid is attached via an ester bond to the beta phosphate of the modified ADP, and on the other side of the pantothenic acid is an activated thiol group, which is the attachment point for acetate. The thiol ester, labeled in the blue 1, has a higher energy than a normal carboxylic acid. Acetyl-CoA is produced from pyruvate, fatty acid, and amino acid catabolism. CoA is used as shorthand to represent the structure of coenzyme A since it's so large. Citrate synthase is the first step. Note that this is a synthase, not a synthetase, since ATP is not used in the reaction. This enzyme catalyzes the condensation of acetyl-CoA and oxaloacetate. This is a highly favorable reaction. The enzyme active site relies on an induced fit so that there is no water interference with the mechanism until hydrolysis is needed. Oxaloacetate will enter first, and a conformation change will occur, which will allow the binding of acetyl-CoA. The enzyme will then undergo another conformational change for catalysis. The enzyme has a catalytic triad of histidine 274, histidine 320, and aspartate 375. A Claisen condensation mechanism is shown in the bottom left as a comparison, with citrate next to it, also for comparison. Citrate synthase is very similar to a classical Claisen condensation mechanism, since the methyl group of the acetyl-CoA forms a carbanion which attacks the carbonyl of oxaloacetate, creating a new carbon-carbon bond. This will create citro-CoA, which can undergo a hydrolysis reaction after the enzyme undergoes yet another conformational change to produce citrate. The thiol ester group being hydrolyzed is extremely favorable and is the driving force of this reaction. Like I said before, carbon-carbon bonds are extremely difficult to make, but using an excellent leaving group like coenzyme A allows this to happen. So the net reaction for citrate synthase is shown here, where oxaloacetate and acetyl-CoA combine together to form citrate. Water is used to hydrolyze coenzyme A, and a proton is also released. Coenzyme A is then recycled for more creation of acetyl-CoA. Remember that for each glucose molecule catabolized previously, that there are 2 acetyl-CoA molecules made. Therefore, this step will happen twice, as well as every other step in the cycle. Step 2 is aconitase, which is named for the important intermediate cis-aconitase. Cis-aconitase is formed via the dehydration of either citrate or isocitrate. The aconitase enzyme will rehydrate cis-aconitase via the addition of water to either one of 2 carbons, which will then form either citrate or isocitrate depending on the position. Cis-aconitase does not typically leave the active site due to the coordination of the iron-sulfur clusters, which position the molecules properly and tightly. The clusters are also used as a signal for the rest of the cell to test whenever iron levels are low. While this reaction is in equilibrium, the reaction typically favors the creation of isocitrate due to the low concentration of isocitrate in the cell. The low concentration of isocitrate is due to isocitrate dehydrogenase, the next step. Isocitrate dehydrogenase is the first redox reaction in the cycle, even though the cycle has 4 steps that are redox reactions. There's actually 2 different isoforms of this enzyme in the cell. They're identical reactions except for one is dependent on NAD+, while the other is dependent on NADP+. The enzyme relies on manganese to coordinate these for catalysis. This step is considered an oxidative decarboxylation since the molecule is first oxidized, followed by the decarboxylation of the CO2 leaving group. CO2, like coenzyme A, is an excellent leaving group since it is highly stable. This is what pulls this reaction and the previous reaction forward. The intermediate of this reaction is oxalosuccinate, which is oxidized but has yet to be decarboxylated. The product is alpha-ketoglutarate, which is very similar in structure to the amino acid, glutamic acid, however it is missing the amine group. The fourth reaction is catalyzed by alpha-ketoglutarate dehydrogenase complex, a set of 3 distinct proteins. This complex is highly similar to pyruvate dehydrogenase complex. It's a highly favorable reaction. In fact, it has enough energy to actually make an ATP molecule, but it doesn't. So instead, it uses this energy and quote-unquote wastes it to pull all of the previous reactions forward and to prevent the equilibrium to favor any reverse reactions. Alpha-ketoglutarate is a building block for many amino acids. This is the final phase of oxidizing the acetate of acetyl-CoA generated by glycolysis and pyruvate processing. The next part will focus on the regeneration of the oxaloacetate. So now that we have gone through the first 4 steps, we have fully oxidized acetyl-CoA, releasing 2 carbon dioxides from the 2 acetate molecules, the 2 carbons from the acetate molecule which entered. Thank you.